Hello everyone, welcome back to another lecture, and this lecture is going to be on enzyme, and we will be discussing about the definition of enzyme, its characteristics. We will also learn that what exactly an active site of an enzyme is. We will learn about coenzymes and cofactors, and then also we will look upon the classification of enzymes. Let's see what enzymes actually are. So enzymes are basically the biocatalysts. What are biocatalysts? So these are the catalysts present inside the biological bodies. And now if we talk about catalysts, that what exactly catalysts are the molecules that are responsible for the conversion of reactants into products. And in this conversion, the catalyst itself does not undergo any change. So what I mean to say is that the catalyst is not having any change in its structure or its form, but rather it is just acting as a medium, or it is providing the surface for the reactants to come and produce products. So these kind of catalysts which are present inside the biological bodies, that is the animals, plants, humans, microorganisms, those are known as enzymes. So enzymes in the biological bodies are responsible for increasing the rate of biological reactions. Without enzymes, it would have been very difficult for the pathways to take place, for the reactions to take place inside the living bodies, and hence the procedures, all the processes inside the bodies would have been halted. Now, in case of the biological bodies, the reactants are known as substrates. So wherever I will be calling substrate, it means that I'm talking about the reactants. The enzymes, therefore, are responsible for the conversion of substrates into products. And these enzymes, they are protein in nature. They are made up of amino acids. So different amino acids, they join together to form long chains, and then they coil up to form the tertiary structure. And that is the active conformation. And this is a protein only. And the enzymes, they are specific in nature, which means that each enzyme will have a corresponding substrate with which it will bind. So every substrate will have a specific enzyme with which it can go and bind and produce products. Not every enzyme can bind to every substrate, and not every substrate can bind to any enzyme. There needs to be specificity between the substrate and the enzyme so that they bind to each other and ultimately produce the products. The enzymes are sensitive and labile to heat. Why? Because you know that the enzyme, they are proteins and they have a native three-dimensional conformation. And this three-dimensional conformation can be altered if heat is applied. In case of increased temperature, this three-dimensional structure can be lost. The conformation will be lost, and hence the enzyme will not be active. So in order to enzyme to function, it has to be in its native 3D conformation. The enzymes, they are water soluble. Now, whatever the molecules you will read in the biological systems, if it is having A's at the end as the suffix, it means that it is an enzyme. So all the enzymes, they have A's at the end. For example, if we talk about lactase, lactase is an enzyme that is responsible for converting lactate into glucose and galactose. The enzymes, they function under strict conditions of temperature and pH in the body. So if there will be an alteration in the pH or temperature inside the body, these enzyme activity will be halted, or it will be affected to some extent. Why? Because again, it has to do something with the active conformation. So the active conformation of the enzyme is stable at one specific pH and temperature inside the biological bodies. If there will be a shift in that temperature or pH, the conformation will also be affected, and hence the activity of the enzyme as well. Now let us understand that how exactly the reactions are taking place, how exactly these substrates are converted into products. Now, the concept of active site comes into play here. Each of the enzyme that we are talking about has an active site present on its surface, which is responsible for converting the substrates into products. Now, what I mean to say here is that not all of the enzyme, the whole enzyme structure is not participating in the conversion of substrate to product, but rather only a specific segment of that enzyme structure is participating in that. And that specific surface is known as the active site. So active site of an enzyme is the region that binds to the substrate and converts it into the product. For example, if you look here in this figure, this purple colored one is the whole enzyme. But if you see this specific segment, which is having a cleft kind of thing, a hole kind of thing, that is the active site. And the shape of this active site should be complementary to the substrate, which is going to come and bind to this active site. Only if the complementarity is existing, then only the substrate will bind to the active site. Otherwise, it will not bind. And this is usually a relatively small part of the whole enzyme. So different amino acids from different parts of the polypeptide chain, they come together and form this cleft kind of structure and hence form the active site to which the substrate binds. Now the substrates that come and bind to the active site, they must be binding with the active site through some forces. Now, those forces are weak forces. Now, let us try to understand it, why they have to be weak forces. Because if these forces were strong, then the binding between the substrate and the enzyme active site would have been very tight, leading to the inability of the substrate to convert into the product and finally release out of the active site. So these forces need to be weak enough so that it can come and fit inside the active site, do the reactions, and ultimately release as product. So that is why they have to be weak forces. Now let us talk about the nomenclature of the enzymes, that how exactly we have been able to categorize these enzymes so that we can understand that what exactly is the function of this enzyme. So according to the IUBMB system, each of the enzyme starts with EC, that is the enzyme class, followed by 4 digits. Now, for example, let's see this example, EC2711. Here, EC is referring to enzyme class. The first digit of this number is referring to the class. So the classes we are going to see in the coming slides. So second class is basically transferases, for example, here. The next digit is referring to the subclass. For example, if we take glucose 6-phosphotransferase, let's suppose that we are taking the example of this enzyme. This is having the digits allotted as EC2711. So 2 is referring to its class, which is transferase, which means that this enzyme is responsible for transferring certain groups. The next digit here, 7, it is referring to the subclass. And what is the subclass? Phosphotransferase, which means that it is transferring the phosphate group. Next digit, 1, it is the subclass, which means that it is going to tell that where exactly this is transferring the group. So it is transferring it to the OH group, which is the acceptor molecule. And the last one is providing the particular identity to that enzyme. For example, here in case it's 1, so glucose binds the phosphate group. So if we can understand that what exactly is the significance of each of these digits, we can go and see in the tables and we can understand that what exactly this particular enzyme is doing. In this example, this is a transferase enzyme, which is transferring the phosphate group, which is being accepted by the OH group, and where it is going to the glucose. So glucose binds the phosphate group and forms glucose 6-phosphate, as you can see here. Now, what are coenzymes? So coenzymes are something to do with the enzymes. For example, you see in the movies, there are stars and there are some co-stars. Co-stars are the ones without which the star cannot function. So co-stars need to be there. Similarly, in case for the enzymes to work, coenzymes are needed in certain cases. Also, there is one more molecule called cofactor. So cofactors and coenzymes are helping the enzyme for its function. And some of the enzymes, they cannot function at all without the presence of these molecules. Now, a complex enzyme contains a protein part called the apoenzyme and a non-protein part called the coenzyme. So this is the protein part, the one that we have been discussing so far. Now with this protein part, there is this non-protein part. When it comes and attaches, in this case coenzyme, this becomes a hollow enzyme, which is the active enzyme. So hollow enzyme, if you read anywhere, it means it's talking about the active enzyme, which has the coenzyme attached in close proximity with the protein part, that is the enzyme. So enzyme binds to cofactor or the coenzyme and forms the hollow enzyme. Now, what is the difference between coenzyme and cofactor? So basically, coenzymes are the organic molecules and cofactors are the inorganic molecules. And inorganic molecules may be some ions. For example, if I talk about the enzyme hexokinase, which is responsible for catalyzing the first reaction of glycolysis, where glycolysis is converting into glucose 6-phosphate. So this hexokinase, it requires one cofactor, magnesium ion, for its activity. In the absence of magnesium ion, hexokinase is not able to function and convert the glucose to glucose 6-phosphate. Now the term, one more term is here, prosthetic group. What is a prosthetic group? It is the term given when the coenzyme tightly binds to the apoenzyme. So when there is a tight association of this coenzyme with the apoenzyme, we call it as a prosthetic group. Here, the coenzyme molecule is the prosthetic group. There are 6 classes of enzymes with which we have categorized all the enzymes to their specific functions that they do. And these include oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Now, you can memorize this by memorizing OTHLIL. So you can use this mnemonic to understand that these are the 6 different classes of enzymes. So we will be now seeing each of these classes one by one. So the first class is oxidoreductases. The name itself is suggesting that this has something to do with the oxidation reduction reactions. So in the oxidation reduction reactions, the electrons are transferred. So the enzymes that are responsible for the transfer of electrons or doing the oxidation and reduction processes, those are oxidoreductases. And the important example is dehydrogenase enzyme. So there are a lot of dehydrogenases which are acting in different reactions of several different pathways. So these are the oxidoreductases. We have one example, pyruvate dehydrogenase, which is responsible for converting pyruvate to acetyl-CoA in the linking reaction. So for example, let's suppose that we have A and this B. So when this is going on, if this kind of reaction occurs, then that is termed as oxidoreductase. The next is transferases. What are transferases? Transferases are the enzymes that are responsible for the transfer of groups. For example, that we just saw in the nomenclature part. So transfer of groups is done by the transferase enzyme. The examples include hexokinase, which is transferring the phosphate group from the ATP molecule and transferring it to the glucose and converting it into glucose 6-phosphate. So for example, let's suppose we have this A, B, and this reacts with C. Now there's something that is this enzyme transferase comes and it transfers the group from A to C. So the final product can be something like this. So that is what the transferases are doing. The next is hydrolases. The hydrolases enzymes, they're responsible for the hydrolysis reactions, and it usually involves the transfer of functional groups to water. And one of the important examples include chymotrypsin. So what is being done? Let's suppose that these are the reactants, the water reacts with it, the involvement of water is there, and there is a transfer of the group B from A to water, something like this. Then we have lyases. Lyases are the enzymes that are responsible for the breakdown of the specific molecules. And one of the important examples include conversion of fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate and dihydrogenase acetone phosphate. So what exactly is happening? This is a 6-carbon molecule, and lyases, specifically here, if we talk, it is aldolase. Aldolase is a lyase enzyme, and this is converting the 6-carbon fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate, which is a 3-carbon molecule, and DHAP, which is again a 3-carbon molecule. So I can illustrate it something like this. Let's suppose that this is the molecule. Now it is being broken down, and the groups that were attached to these, they are separated. The next is isomerases. Isomerases, these are the enzymes that are responsible for the isomerization of the molecules. And the important example includes conversion of glucose 6-phosphate to fructose 6-phosphate in the glycolysis pathway. So what is happening here? For example, let's say this is the molecule. Here, X is attached to A and Y is attached to B. And now isomerization happens where X is replaced by Y and Y is replaced by X. So the chemical formula is remaining the same, but there is a difference in the structure of the molecule. So that is isomerization here, and isomerases do this thing. The last one is ligases. Ligate means to join. So ligases are the enzymes that are responsible for the joining of the molecules. So there is a ligation happening here. And these reactions are usually coupled with the cleavage of ATP. For example, that there is the molecule A and there is a molecule B, and these are joined together to form one single molecule AB. So this is something which is being done by the ligases. So these were the 6 different classes of enzymes. Now, lastly, let's just see that what are the functions of enzymes. So in the absence of enzymes, biochemical reactions will hardly proceed. It might take millions of years to proceed without the enzymes. So enzymes are very important. The enzymes are basically increasing the rate of reaction by up to 10 the power 7-fold times. So that's a lot. So the reactions that might take millions of years can be done in a fraction of a second. So that is the power of enzymes. So therefore, they are very crucial for the metabolic pathways and also the living organisms. Now, there are certain medical applications of the enzymes. For example, the enzyme-related disorders can be treated with the enzymes in the industries. For example, there is a lactose intolerance in the human beings, you see. So why it is happening? Because these humans, they are deficient in an enzyme called lactase. So if lactase is not there, they are unable to tolerate the lactose. So this comes in play there. And they also assist in metabolism. They can be used for the drug delivery. They can be used to diagnose and detect the diseases, and also in the manufacturing of several different medicines. It also have a lot of industrial applications, such as the enzymes amylase, lactase, cellulase. These are all enzymes which are used very widely in the industries for the breakdown of complex sugar into simple sugars and produce products which are commercially very important. Enzymes like pectinase, they are used to break the hard pectin, which are used in the fruit juice manufacturing. Lipases, they are also used to break down the lipids in the industries, maybe to remove the grease stains. And the enzymes, they're used in the detergent and washing soaps as well. And protease enzyme, they're used to remove the stains of the protein nature, like blood and sweat, etc. So apart from these, there are several different other important functions of enzymes inside the living bodies, which is very, very important. And without the enzymes, these functions cannot take place, and hence the human being can die as well. So that was it about enzymes. Thank you so much.